throughout the natural selection, we and other organisms, including bacteria, produce only gene product that's necessary for the cell so that we conserve the resource, biological resource. And the production, certain production of enzyme is regulated by the feedback inhibition or gene regulation. And we're going to have one example or multiple example of gene regulation in bacteria because those are well studied. So those are called operon model. So here's the example of regulation of the gene and the pathway by feedback inhibition and the regulation of gene expression. So let's see. So this example is the purpose of this pathway is to produce tryptophan. And we have three enzymes that catalyze the initial substrate. So this is initial substrate, it gets catalyzed by these three enzymes. And this enzyme are multi-subunit enzyme composed of two different proteins encoded by two different genes, TRPE and TRPD for the enzyme 1, TRPB and TRPA for enzyme 3. Right. So if you have the pathway that produces tryptophan, if you want to produce, you need to transcribe these genes from the DNA to RNA, and it gets translated into the enzyme. So this is the gene regulation of expression. And once you have a production, and if you have enough tryptophan, you do not want to waste all your resource for production of tryptophan, and if you have too much tryptophan, that doesn't do anything much, and you want to put the limited resource to something else, for something else. So that's why you want to shut down the mechanism, shut down the system. So there are two mechanisms. One is feedback inhibition. So tryptophan, the end product of this pathway, can inhibit the first enzyme and stop the production. Second, this end product should also stop the regulation, stop the transcription of these messenger RNA from the gene, so that you do not produce enzyme 1, 2, 3 if you're not, if you don't really need it. So in bacteria, this is done by the operon, the system called operon, which is a whole bunch of sets of genes in the same pathway with a single on-off switch. And this switch is called operator. And the operon contains operator, which is a switch, promoter, which is required for the transcription of the messenger RNA, and multiple genes. And the operator, some operator is repressor. So the repressor binds to the DNA, the operon, and block the RNA polymerase, which is necessary for the transcription of the RNA. So if the repressor binds to the operon or operator, then it stopped the transcription. And this repressor usually are the product or separate regulatory gene. So how do they look like? This is how they look like. This is tryptophan operon. You have regulatory gene, which is usually somewhere other than away from the tryptophan operon. This encodes a protein which is repressor. But in this form, it is inactive, so it doesn't bind for now. So, in the absence of this repressor, the RNA polymerase bind to the promoter of operon, and the RNA polymerase will synthesize the messenger RNA, transcribe all those genes into the messenger RNA, and these get translated. So this gene, this single messenger RNA has multiple genes on it, and creating the enzyme. So two protein that form enzyme 1, enzyme 2, and these are the enzyme 3, necessary for the tryptophan synthesis. When you have too much tryptophan, you want to turn off. So now you have repressor in the presence of excess tryptophan, and the tryptophan binds to this repressor, induce the conformational change, the three-dimensional change of the shape of this repressor, result in the repressor that can bind to the operator portion of the tryptophan. Now this promoter is blocked by this repressor, and the RNA polymerase cannot bind to and proceed the transcription. So this way, the presence of excessive end product can stop the synthesis of all those genes at once. And by doing so, they can conserve the energy when they don't need the tryptophan. So let's reiterate the important concept. The operator is the stretch of the DNA sequence that's in front of those many genes. Operon is this large unit of the DNA that includes operator, promoter, and the multiple genes. The repressor is the gene product of the regulatory gene. Here, in this case, it's TRPR. And yeah, this is a regulatory gene, and the co-repressor is a tryptophan in this case. Something that binds to the repressor so that they function as a repressor together with the real repressor, that's a co-repressor. And that binds to the operator of the operon, making the RNA polymerase not to proceed the transcription of those multiple genes. So this tryptophan operon is the example of repressive operon. It's usually always on, and in the presence of co-repressor, it gets repressed and stop the transcription of all those genes. But there are some other mechanism called inducible system instead of repressible system. So compared to the repressible one, the inducible one is usually off. And so in this example, we have a lac operon, which is encoding the enzyme that you catalyze the lactose. So in the presence of lactose, the bacteria can utilize the lactose. But if you don't have lactose, why do you waste your resource creating the enzyme that catalyzes the lactose? So they need a regulation for the lactose digesting enzymes. So in this case, lac operon has lac regulatory gene right in front of the operon. So this gene is transcribed into messenger RNA and translated into the protein. Unlike the tryptophan, this repressor is active without anything. So it can bind to the LAC operator, which is in the part of the LAC operon promoter. And RNA polymerase cannot proceed because of the operator inhibiting the polymerase going into the gene. So no transcription of those genes usually. But in the presence of allolactose, this is a substrate, then they can sense the presence of the lactose, and if they have enzyme that can digest, and we can use those as an energy source. So in the presence of allolactose, it binds to the repressor. Now it's inactive. So they cannot bind to the LAC operator. Now RNA polymerase is free to go. They can transcribe all those genes, and they get translated into those beta-galactose, pentamase, and trans-acetylase. So these are necessary for the digestion of lactose, and it will be used for the bacteria for the energy source. So in the tryptophan operon system, we had tryptophan as co-repressor. So co-repressor bind to the repressor, activate and stop the synthesis of the gene necessary for the tryptophan synthesis. But in inducible system, we have inducer instead of co-repressor. So inducer in this case is the lactose that binds to the repressor, inactivates the lactose repressor, activate the transcription of this multiple genes necessary for the lactose digestion. So generally speaking, inducible system is found in catabolic pathway, such as the use of lactose for the energy source in bacteria. The repressive operons are usually found in anabolic pathway, which is the synthesis part. So in this case, we're discussing the synthesis of tryptophan from simpler molecule. That was repressive operon, and in both cases, in the tryptophan and LAC operon, in catabolic and anabolic pathway, active form of the repressor binds to the operator and switch off many genes expression in the same time. So we study how the repression happens even in the repressive operon and inducible operon. But what drives this RNA polymerase binding and initiation of the transcription? So let's see the positive regulation rather than negative feedback regulation of the gene transcription. So E. coli can use glucose and lactose. But when both of them are present, they want to use glucose instead of lactose. Glucose is a regular normal substrate for glycolysis, and enzymes are present. It's rather cumbersome and require additional resource to digest the lactose by turning on the LAC operon and turning on and synthesizing those LAC digesting enzymes. So if they have glucose, they don't want to make the lactose. Even though they have lactose, they don't want to make the lactose enzymes. But when they have no glucose around, they have to use the lactose as the alternative source for the energy. That's why cell has to sense the lack of glucose, low level of glucose. So if you have low level of glucose, there's a small molecule accumulation that's called CAMP, cyclic AMP. So when you have a low glucose level, CAMP accumulates, and there is a protein called CMP receptor protein. Oh yeah, C. It's a straightforward name. CRP binds to the CAMP. And this binding of CAMP to the CRP changes the three-dimensional structure of the CRP. And now this CRP is active. It can bind to the lac promoter. And by binding to the lac promoter, it recruits the RNA polymerase. It increases the affinity of RNA polymerase to this lac promoter. So by doing so, it recruits this polymerase, RNA polymerase. And that in turn drives the transcription if you have inactive lac repressor. So in the presence of lactose, then you have LAC-Z and other genes expressed. This is active regulation, I mean positive gene regulation. But if you have, even in the presence of lactose, if you have enough glucose around, the cell wants to do the glucose instead. So now in the presence of glucose, you don't have CMP accumulation. So CRP is no longer active. It doesn't bind to the lac promoter. So it does not recruit the lac promoter, even though you have a lactose available. So this is a very simple model, but even this simple lac promoter, you can have positive gene regulation and negative gene regulation at the same time. The gene regulation. So this shows that the gene regulation is very intricate, complicated, and works really well.